Good morning, students. We are going to study dry chemistry analyzer. So it's one of the analyzer. We have studied many analyzer, and now the last topic in automation we will study about the dry chemistry analyzer. Let's know some introduction about this special type of analyzer. Here are some example of dry chemistry analyzer, like bedside glucometers, cholesterol and triglyceride analyzer, hemoglobin analyzer, and blood group analyzer, a blood group reader also. So these are the the analyzer which are available in the market and hospital. So glucometer, I think you might have seen everyone. So glucometer, which we can put in keep in our home also, and there are some strip is available on which we put some one drop of blood, and after that the machine, the glucometer, gives the glucose values. Okay, so bedside glucometer. Similarly, cholesterol and triglyceride analysis only gives the result of cholesterol and triglyceride. Hemoglobin analyzer, which gives the value of hemoglobin only, and blood group reader is the the analyzer which gives the reading a result of blood group. Okay, either A positive, B positive, or O positive, whatever the blood groups. So the blood group reader can give the the blood groups. So these are the one few examples you have studied for dry chemistry analyzer. This is also known as dry chemical analyzer also. We can write it, okay? But in the syllabus, as a nomenclature is given dry chemistry analyzer, so no problem. You can write. You can know by dry chemistry analyzer also. Why we call it dry chemistry? Because there are some dried reagents are there, and that dried reagents will react with the sample, and then finally it produce a color. And the intensity of the color we can measure it, and it gives the the results. The principle of the method is the analyzer is how this analyzer works. So let's know about this. Dry chemical method utilize reagent slide that are composed of several layers. So here we have there's some chemicals which are dried, right? Some chemicals which are dried. Uh, you can consider the reagents which are used in the laboratory procedure. Let's say the estimation of glucose, whatever the reagents we use. So the reagents contain some chemicals which are dried on the slide, okay, and they're composed of several layers. There are several layers present on the slide. What are the several layers? The first layer is a spreading layer. Second layer is a scavenger layer. Third layer is a reagent layer, and fourth layer is a support layer or plastic layer. So there are four layers, on the slide. On the dry chemistry method, this dry chemical analyzer uses a special slide that contain four layers. Okay, spreading layer, scavenger layer, reagent layer, and plastic or support layer. So let's know one by one what happens in these specific layers on the slide. Okay, now we just look on this image. This image, you can see at the top we uses sample drop which is placed on the spreading layer, right? This is a spreading layer which has a sample drop first. Then the second is a scavenger layer just below the spreading layer. Third layer is a reagent layer, and the fourth layer is a support layer. You can see the different layer present on the slide. Yes. Now next is the what it is showing, the figure showing various layer in a dry chemical analyzer. Okay, and this is the principle of detection of reagents. What is the principle of detection of reagents? That when the sample is added to a spreading layer, then it goes to the reagent layer through the scavenger layer, right? And after that it produces a color, and we can measure the intensity of the color here. Okay, and we can display the result, right? So let's know one by one what is the spreading layer means. The spreading layer is an area for aiding sample or control or standard. So this is an area, right? These are the spreading layer is the layer or the the place where we add sample or we add control or we add standard, right? What happens in scavenger layer? Scavenger layers allows selected components to filter through and penetrate through the reaction layer, or what we say reagent layer, which in turn activates the dehydrated reagents. So this is what the scavenger layer is acts as a, the sun like a filter. Okay, it acts as like a filter, and that filter allow only selected component to pass through. Let's say you are doing the glucose test, so it will only allow the glucose to pass. Or if you are doing the protein test, it will only allow the protein to pass. Okay, so the selected component is passed and penetrated through the reaction layer or reagent layer where reaction takes place. Okay, because the sample component, the reagents, we can when they are allowed to mix together, then reactions takes place, and after that it turns into the color product, right? So reagent layer, what is reagent layer? It contains lyophilized or dry enzymes and buffers which are necessary for analysis of a specific analyte in the sample. So what we usually do in the wet lab, in the our lab, we manually what we do, we take reagent, we take sample, we mix together, and we do in mixing, and then after that we do incubation, and after incubation we take the OD of with the help of colorimeter, right? So this is the what manually we do in the lab. So that reagent that what we take in the lab. So here the reagent is basically all the chemical component of the reagents are here taken in the reagent layer. Okay, so reagent layer contains lyophilized or dry enzymes. So the reagents are basically dry form. It is not liquid form. It is dried form. Okay, all the reagent component, even the buffer components, so they are in the dried form, and they are all the reagent components which are necessary for the reactions are present in the reagent layer, right? I hope you are understanding. And in the support layer, this is the plastic layer made of plastic, which provides the support to the whatever the layer you have studied above, spreading layer, scavenger layer, or reagent layer. So those support layer provide support to the whatever the layer you have studied. Okay. So what is the role of support layer here? It allow light to pass through and is directed through the reagent layer. Okay. So this is the layer which allow to take the reading. It allow to take the absorbance. Understood? So this is what we understand. Yeah, Nand Kishore has given one of the uh, one of the slides, one picture. Yes, we'll talk about this. So I hope you have understand the support layer. What is the role? It allow light to pass through, and which is directed through the reagent layer, and then it come back to the the colorimeter as towards the detector. So when the detector detects that how much light has been reflected and how much light has been absorbed by the color substance. So based on, so here more color, more OD will be there, and more OD means more absorbance. If there is more color, more absorbance you will take, you will find, and after that, when there is a more absorbance, so accordingly we can say there is a more concentration of the component in the given sample, right? So this is the what the principle, the working principle of the dry chemistry analyzer you have studied. So here a chemical reaction is initiated to produce a color in the reagent layer. This is the concept you have to make, that the reaction is only possible when you allow the sample to react with the the chemical component of the reagent layer. Okay, and the color is produced. So when light is passed through the support layer or plastic layer, so it passes through the color reagent, and finally it hits the white spreading layer, and this will cause some of the light reflects back through the reagent layer to a photocell. So photocell is a detector which detects that how much light has been reflected from the reagent layer. Okay, where we get reactions. So The amount of reflected light is indirectly proportional to color intensity, because here the the light which has been reflected, so it decreases when there is a more absorption, right? So if there is a more absorption, so the reflected light will be, intensity of the reflected light will be less. So the amount of reflected light is indirectly proportional to the color intensity. So less the intensity of the reflected light, more will be the the color of the what we say more will be the color of the reaction mixer. Okay, so reaction mixer will absorb more light, and accordingly the light which has been reflected will be lesser. So the photo detector will detect the reflected light, and based on that we can give the results. This forms the basis to determine the concentration of the analyte. This is the basis. This is the basis that in this basis we can analyze any component present in the sample. So you have to provide the specific reagent in the reagent layer, and accordingly we can be able to determine the specific component present in the the sample. So I hope you have understand the principle. And now, what are the advantages of this dry chemistry analyzer? What are the advantages? Here, the storage requirements for reagents are minimal, since no wet reagents are required. This is the very good advantage, that we do not require to store the reagents in the liquid form, because when we whatever the auto analyzers you have studied in the previous classes, in all the analyzer they utilizes the wet reagents, liquid reagents. So there should be the storage system that should store the large volume of the reagents, right, in the liquid form. But here we do not require reagent storage systems, because the reagents are coated on the slide as a reagent layer. Okay, so this is what the space can be minimized, right? And no pipetting steps are needed. We are also like reagents. When we talk about the analyzer, what we studied in the discrete form or continuous form or centrifugal analyzer. So in all the form we have studied that we need to pipette the specific volume of reagent, specific volume of sample. But here we do not require to pipette out the reagents, because the reagents are available in the dry form on the slide. Okay, no sample dilution is required. Here this is the one big advantage, that sample dilution is not needed. And 10 or 11 microliter of sample per test is used. So what we understand here, that the sample requirement is also very lesser in compared to what we studied in the previous different types of analyzer, right? It only require 10 or 11 microliter of sample. Okay, and from that sample quantity you can be able to measure the specific component present in the sample. If you want to do the glucose test, it can give glucose test results. If you want to do triglyceride test or cholesterol test, so all whatever the test you are doing, so it requires only less amount of sample, right? So we have come to the end of the slide. Now if you have any question, now we can discuss over here. Yes. uh, let's discuss any doubt you have.